Hi team, welcome to our video about nucleic acids. We're going to be talking about DNA and RNA and the monomers, nucleotides, that make up both DNA and RNA. So in this first picture here, I'm showing you DNA, the double helix, which is often found wrapped up in these much larger structures called chromosomes. And in our cells, most of our cells contain DNA within the nucleus of the cell. Now, of course, prokaryotes like bacteria also have DNA, but they don't have a nucleus. So we're just going to be talking about the larger structures of DNA and RNA and their monomers, nucleotides. So here's a general structure of the nucleotide monomer. If this is a monomer, that means DNA and RNA are polymers. And the three components of this nucleotide monomer are the phosphate group, the sugar group, and the nitrogenous base. And we'll talk more about the sugars and bases in a minute here. We have talked about phosphates a little bit when we talked about lipids. Phosphates contain a phosphorus and some oxygens, and they're negatively charged. And phosphates giving DNA this negative charge, also RNA, the negative charge, is a really important part of what helps DNA and RNA form their structure within the cell. So phosphates within the backbone of the nucleotide give a charge to nucleic acids. Next, let's talk about the sugar component of the nucleotide. We've got ribose and deoxyribose. They're both carbohydrates, sugars in their ring form. And there's one important difference between ribose and deoxyribose. That's this OH versus H on the second carbon here. Deoxyribose has its name because it has one fewer, it's had an oxygen removed, it has one fewer oxygen than what's present in ribose. So deoxyribose is what makes up the backbone of DNA, deoxyribonucleic acid, whereas RNA is ribonucleic acid. Okay, so these the differences in the sugar of the RNA and DNA nucleotides are not very dramatic, but the presence of this extra oxygen on the ribose sugar means RNA is a little bit more unstable, or it's less stable. So RNA is less stable because it's got this extra oxygen group. DNA is more stable because it is deoxy. Last but not least, let's talk about the base, the nitrogenous base that's part of nucleotides. There are two different types of nitrogenous bases that we'll talk about, purine and pyrimidines. And you can see that in general, there are two types of structures as well. We've got the two-ring structure. It's got a 6-member ring and a 5-member ring, and that's called a purine. And then we've got another 6-member ring, a single ring, called a pyrimidine. Purines, I like to remember the name of purine as the small name with the big structure. Pyrimidine has the great big name with the much smaller structure. Okay. There are two purines which are present in both DNA and RNA, and that's adenine A and guanine G. You can see both adenine and guanine have two rings. They have the general purine structure. And then pyrimidines, there are three different pyrimidines that we'll talk about. Cytosine C is present in both DNA and RNA. And then thymine T is present in DNA, but uracil is only present in RNA. So T and U swap out depending on which polymer, DNA or RNA, is present. Terrific. How can we remember, though, that cytosine, thymine, and uracil have the shape of a pyrimidine? Some folks like to remember CUT, cut the pi. And then over here for purines, we've got pure Ag, purine, adenine, guanine. I should say pure as gold. Pure as gold for purines, adenine, and guanine, and cut the pie for pyrimidines, C, U, and T. So let's look now how all three of these components come together in a nucleotide. We've got the thymine base attached to the 1 carbon of this deoxyribose sugar. And then the 5' carbon, carbon number 5, is attached to the phosphate group. So that's why this is referred to as the 5' end, because we've got the 5' carbon attached to the phosphate group that's exposed on this end. And then on the opposite end of the molecule, we've got the 3' end. The 3' carbon, in this case, is attached to an OH group, and this OH group is where the next nucleotide will be added. It binds, but a phosphate from the next nucleotide will bind here to this OH group. So you can imagine this O was formerly part of an OH group right up here on this 3' carbon, but now the hydrogen is gone. And a phosphodiester bond has formed between these two molecules. So the bond that holds two nucleotides together is called a phosphodiester bond. And it's formed, like so many other polymers, by a condensation or dehydration reaction, linking together the 3' end of one nucleotide to the next one. So we could draw another thing that's going to come in, right there. That phosphate group on the 5' carbon is going to come in and attach to the 3' end of this molecule. The 5' end up here is not going to add any new bases. It's sort of the beginning. Whereas the 3' end is open for business and it can add new nucleotides down there. Terrific. Now, that previous structure we were looking at was a single-stranded DNA molecule, but we usually think of DNA as being double-stranded. So let's talk about how purines and pyrimidines join together to form double-stranded DNA. Now, this bottom image here is showing how a purine and a pyrimidine typically pair together with hydrogen bonds. And depending on the pairing, it's 2 or 3 hydrogen bonds that keep those bases bonded together. And having a purine and a pyrimidine ensures that the DNA double helix holds strong at 20 angstroms in width. If we had 2 pyrimidines, you'd see the DNA double helix sort of get really narrow and skinny. Whereas if we had 2 purines, it'd have a really wide double helix, and having either of those present would mean a really wibbly-wobbly molecule. If we always have a purine and a pyrimidine, then we're ensuring that the double helix stays strong and sturdy at a uniform width, in this case of 20 angstroms. So the pairings between purines and pyrimidines that we see, here's a purine, adenine pairing with a pyrimidine, thymine. So A and T pair together with 2 hydrogen bonds holding them together. And thymine, of course, would be in DNA. We might also have uracil if this were an RNA molecule. And then we've got our purine, which is guanine, and the pyrimidine, cytosine, which are held together by 3 hydrogen bonds. Now, the consequence of having 3 hydrogen bonds for guanine and cytosine is that if we have a long stretch of G's and C's in the DNA, the complementary strand is going to have 3 hydrogen bonds between each of these pairs, it's going to take a lot more energy to break this stretch of DNA apart than something of the same length that's made up of T's and A's. Because there's only 2 molecules holding, or 2 hydrogen bonds rather, holding each of the T and A molecules apart. So having a stretch of these triple hydrogen bonds is going to be harder to break apart or denature the double helix if you have many G's and C's. And here, 2 hydrogen bonds in the A and T pairing means it's a little easier to denature the double helix. A little bit less energy is required to denature that stretch, forming 2 single-stranded pieces of DNA. All right, the last picture I want us to look at is this fully double-stranded nucleic acid molecule. And you can see on the left-hand side here, we've got our phosphate group linked to the 5' carbon. And then at the very bottom, we've got this free OH from the 3' carbon, ready to add a new nucleotide on the bottom here. So 5' to 3' direction. And each of these phosphate groups is forming a phosphodiester bond. Here's the diesters to hold the 2 nucleotides together. But now if we look at this top molecule, T, it's got 2 hydrogen bonds holding it together with its partner A. G is paired with 3 hydrogen bonds to its partner C. Let's look at that, that partner strand of DNA. Notice that the 5' carbon, the phosphate group, is way down at the bottom on this other strand. And the 3' OH is up here at the top of the page. This second strand is running anti-parallel to its partner. So we've got 5' to 3' on the left, 5' to 3' on the right, but they're opposite one another. They're anti-parallel. And it's in that anti-parallel fashion that we see the bases pairing up and then the DNA backbones relative to one another. Great. I also want to point out that this is a DNA molecule, a double-stranded DNA molecule, and not RNA for 2 different reasons. The first reason is we've got thymine, not uracil. If it were RNA, we would have a uracil here paired with adenine instead. And the second reason is we've got deoxyribose. So if we look at our 3' most sugar group down here, you can see we've got 1 OH group, and then here we have no OH. It's been removed, right? We've got deoxy sugars. Each of these sugars is missing that second oxygen. This is deoxyribose. It's missing that second oxygen. So it's not ribose, it's not RNA. Now, the fact that this molecule is double-stranded doesn't really tell us whether it's DNA or RNA, because RNA can also be double-stranded. All right, so that's a summary of nucleic acids. Feel free to watch as many times as you want. Bring questions to class, to office hours, and to your SI leader. See you on Monday.